Scientists discovered a new class of anti-malaria antibodies that could help us tackle one of the world's deadliest diseases. Researchers at the National Institutes of Health have identified these novel antibodies, which target a previously unexplored region of the malaria parasite, offering a potential breakthrough in malaria prevention. One of the newly discovered antibodies demonstrated significant protective effects against malaria parasites in animal models. These antibodies bind to areas of the parasite not targeted by current malaria vaccines, opening a new avenue for combating this persistent global health threat. Malaria, caused by Plasmodium parasites transmitted through infected mosquito bites, remains a major public health challenge. While rare in the United States, it had a devastating global impact in 2023, with the World Health Organization estimating 263 million cases and 597,000 deaths, primarily among young children in Africa. Plasmodium falciparum is the most common and deadly species, making the need for effective countermeasures critical. Of the 5 Plasmodium species that cause malaria, Plasmodium falciparum is the most common and deadly, particularly in Africa. Recent malaria intervention advances include new vaccines for children in endemic regions and promising anti-malarial monoclonal antibodies, which target the Plasmodium falciparum sporozoite, preventing it from infecting the liver and developing into blood-stage parasites. Existing malaria preventions target a specific part of the parasite, but this research looked for new targets. They found a promising antibody that attacks a different spot, potentially offering better protection, especially for infants. Scientists discover bacteria that eat forever chemicals, offering a potential breakthrough in combating these persistent pollutants. A recent study revealed that a bacterial strain called Labris portucalensis F11, isolated from contaminated soil, can break down the exceptionally strong carbon-fluorine bonds in PFAS, including some of the concerning shorter-chain varieties. PFAS, or per- and polyfluoroalkyl substances, are a group of man-made chemicals widely used since the 1950s in numerous products, from non-stick cookware to firefighting foam. Their widespread use and resistance to degradation have led to their ubiquitous presence in the environment and even in human blood, earning them the moniker forever chemicals. While most remediation efforts focus on containment, F11 bacteria can dismantle these chemicals. Within 100 days, the study showed F11 metabolized over 90% of perfluorooctane sulfonic acid, a hazardous form of PFAS. It also degraded significant amounts of other PFAS compounds. This research tracked not just the parent PFAS, but also the resulting metabolites, some of which F11 further degraded. This is crucial, as some byproducts are equally or more toxic. While degradation is currently slow, future research will optimize conditions for faster consumption, even with competing carbon sources. The long-term goal is a practical bioremediation strategy, potentially using F11 in wastewater treatment or through bioaugmentation in contaminated soil and groundwater. This research marks a significant advance in sustainable PFAS remediation, offering hope for a future with less forever chemical contamination. Scientists discovered a mortality timer within cells that may hold the key to slowing aging and extending lifespan. By studying yeast cells, they found that the size of the nucleolus, a structure within the cell nucleus, plays a crucial role in determining cell longevity. Smaller nucleoli were associated with longer lifespans, while larger nucleoli led to cell death. The nucleolus houses what's called ribosomal DNA, or rDNA, which encodes the RNA portions of ribosomes, the protein-building machinery of cells. As cells age, the nucleolus tends to expand, and this expansion is linked to DNA damage and cell death. The researchers found that when the nucleolus reaches a certain size threshold, it becomes more leaky, allowing harmful molecules to enter and damage the rDNA. This damage can lead to chromosomal rearrangements and ultimately cell death. By manipulating the size of the nucleolus, the researchers were able to delay aging in yeast cells, suggesting that maintaining a small nucleolus could be a potential strategy for extending lifespan. While this research was conducted in yeast, the findings have implications for human health as well, as the underlying mechanisms of aging are often conserved across different organisms. Future research will focus on understanding how the nucleolus regulates aging in human cells and exploring potential interventions to maintain its size and function. This discovery could enable scientists to develop interventions that delay age-related diseases. Identifying the nucleolus as a mortality timer provides a new target for potential anti-aging therapies.